So the modern version of gene discovery, we're still very interested to discover genes and what they do. And we just got an amazingly larger amount of tools available to us. Maybe one of the more important things that we have going for us is that you can study not organisms that are not model organisms. So previously you'd be pretty well restricted within insects to fruit flies or small number of insects that people are studying. But these days you could go out and you could find an organism that you're interested in and do a full genomic investigation if you wanted to. So here's an example of ladybird beetles again, showing some natural variation. There are described subspecies of this group and lots of variation in whether they have spots or not, whether the predominant color is black or more of this orange or red. And we'll talk a little bit about the investigation into what causes these patterns. So maybe on first blush, it doesn't look like there's a lot of reason for these different patterns, but you would have the ability to get in there with some genetic tools and try to figure that out. So it still starts from the same place, still starts from the same desire to investigate the genetic basis for phenotype differences, but the steps in the investigation are a little bit different here. We'll talk about those moving forward. So one of the big advantages of modern genetic investigation is that it's relatively fast and relatively inexpensive to get a whole lot of DNA evidence. So you could do a sequencing of a whole genome, and that'll cost you on the order of thousands of dollars rather than hundreds of thousands of dollars. And you could zoom in and compare, say we're comparing these two kinds of coloration, and find out what genetic differences are consistently different, or at least in the most cases different between these two kinds of beetle. So in this illustration here, this is what's called linkage disequilibrium map. Don't worry about that term. But basically it's a statistical approach to take all the genomes for lots of different individuals and see if when there's a consistent difference in coloration, what parts of the genetics are consistently different, where the dark spotted one would have some set of genetic information that's most often found in dark spotted individuals, and the one without spots, there'd be some genetic information that's most often found in that group. So you'd see this difference. It's often a split, a binary split of yes, has it, or no, doesn't have it. And looking for locations in the genome that have a pattern that matches the phenotypic differences. So this is an illustration of lots of location across the genome, and this red area with the arrow is an area where not only these places, but also some around them, this big red zone of genetic information, locations on the genome that are more consistently different between these different kinds of beetles. So that's a genome-wide investigation. This is an investigation that zooms into a finer scale. So this is still the same kind of plot, lots of dots on this graph, but investigating in more detail. And this region here has a big peak of dots that are showing consistently lots of signal, lots of statistically significant signal of places in the genome that are different between these two kinds of beetle. And so within that, kind of using the information that they might have from a previous study about what's contained within the genome of this organism, they've got a gene here called GATAE. They've got a gene here called PNR, or Penier. Should have pronounced that ahead of time. Genetic information, gene, and the difference between these two kinds of beetle somewhere in this location. So there's differences in length, maybe some extra DNA, maybe some missing DNA, comparing those two. And so this is kind of getting in more detail. You might have an initial understanding that some part of the chromosome was important. And then this gets out of fine scale and says that this is specifically the gene that's important, as in being the basis for this difference, this major difference between two kinds of beetle appearances. So we don't get to do this kind of investigation, but I'm hoping you can kind of visualize it. We'll see some examples, sort of manufactured examples of what you might look at for the kind of data for this. All of that getting to the point of having a candidate gene. Next thing what you'd want to do is to really lock it in and say we have definite evidence that this gene causes this phenotype. And one of the more solid ways to demonstrate genetic causes is to have the gene present, a gene or an allele, a version of that gene, have it present every time that the phenotype is present, and have it missing or a different version every time that the phenotype is missing or a different version. So this was part of the same study where they were locating that genetic location, and later on they did some manipulation. Again, some modern tools available for this kind of approach. And what they did was they artificially removed the ability of these beetles to make that gene product. And every time, no matter where they started from, so they started with these 4 different kinds of wild, naturally occurring beetle forms. And no matter which one they started with, as they did this manipulation to disable the gene, their candidate gene called Pannier, they deleted that. And every time across the board, they got this orange, like flat orange color beetle coloration. So pretty solid evidence. This is a single genetic change in a very controlled laboratory environment, and they get the result of seeing a difference in the coloration. There's more to it, of course. I won't go into details on the rest of the study, but big phenotype change as the result of changing one gene really supports the idea that that is the causative gene for at least part of this phenotype difference. So lots of genomic tools. The genome there is all the genetic information contained within an organism. And there's a lot of it out there. So this is something that was making news a long time ago now. We're at about 20 years in. And 20 years ago, it was a really big deal because they had assembled lots of investigators from around the world to get at this goal of sequencing the human genome. And that's millions of base pairs. Here we have individual chromosomes at 250 million base pairs or so for the longest ones. And that was a lot of effort to get in there and individually determine the nucleotides that make up all these different chromosomes. And as you can see, lots of people involved, and it took lots of time and a whole bunch of money to get in there. So it's sort of the dawn of a new era for genetic investigation, was having a genome and having the ability to get DNA sequence a lot easier for your target organisms. So humans obviously got a lot of attention, and that was the early stages. And maybe I'll jump ahead here. So around 2001, the sequence of the human genome estimated about $100 million of research money to put that together. And time and technology moved forward and things got cheaper, faster, easier. And currently, for about 5 years now, you could sequence a human genome for a lot closer to $1,000 than $100 million. Just kind of bizarre, maybe expected, but pretty stark difference in the amount of money it costs to get this kind of data. So people are generating new human genomes on the regular these days to get at their own particular investigations. And one of the reasons that that's possible is through new technology. And this is a schematic of one of the possible technologies you could use. As a whole, this is all called next generation sequencing. So we don't go into too much detail on how to sequence DNA. We might do some of this in lab. But the technology, basically this next generation sequencing technology, involves grabbing up little bits of DNA, kind of highlighted here. So you grab little bits of DNA, and through an isolated, individualized process, these bits of DNA can be copied and then They're controlled and they're connected to these little lipid droplets, and then dropped into a microscopic tray where they can be individually sequenced. They basically like grow these fragments and determine, as they're adding nucleotides like this C nucleotide, they determine which of these different strands is building with a C nucleotide at any time. And they collect up all that data and they get the sequence for this little DNA piece and this little DNA piece and this little DNA piece, and they end up connecting those all together into one massive collection of DNA data. So more than you need to know, but pretty fascinating science, and things are still getting faster and cheaper and more accessible. So I'm excited to see how things move forward here. So this technology makes it easy to sequence human genomes, but also just opens up the field to every organism that's out there. As I've mentioned, everything that's alive has DNA, and so you could take this approach to investigate plants, fungi, bacteria, whatever you like besides animals. This is a rough estimate. I just went on the Wikipedia page for these. A rough estimate of what number of genomes have been sequenced. So it basically reflects kind of a combination of human bias and natural abundance. So there's more animals being sequenced. There are lots of people out there who are researching various kinds of animals. And then some of these groups a little bit less abundant, the algae, the so-called protists, fungi, not a lot of attention to them. Oh my gosh, I don't even have plants on here. Fill in the plants. I don't know how I missed those. We'll find the plants on our own here. All right, we're going to Wikipedia, friends. Sequence plant genomes. So all I did for these said mosses and flowering plants and so on, and my estimate was basically to take the number of references here. So 193 references. A lot of these are the titles of papers that are talking about the release of a genome. So I'm going to say there's about 200 of those. Oh my gosh. All right, well now we got plants on there. So lots of new data investigating all kinds of questions. If you've got an organism with something of interest, just for its own sake, or also maybe for how it relates to humans, you can get in there and take these genomic tools to the question. Getting back to humans, we're very interested in humans for a variety of reasons to understand why people are different just on a basic level. We've got appearances, we've also got metabolism and interesting features like lactose intolerance is a metabolic feature, genetic diseases. Some of those are particular to certain populations of people throughout the world. Humans around the world we're one species, and that means, in part, that means that any one human is genetically compatible with a human from anywhere else in the world. So in spite of all this distance that our species has covered, a person from native to South America would still be able to start a family with somebody native whose ancestors were more recently in Africa. The separation between any two of these was about 70,000, 50,000 years ago, the so-called out of Africa. So the ancestral population of people that moved away from Africa, the original place for human species, various times on here as they got into different parts of the world. So this graph tells you, this chart tells you a few different things, tells you, when people got into different areas. Kind of interesting that this matches very well with archaeological evidence. So for example, the first evidence of human activity in North and South America tracks pretty well to this estimate that you can get using DNA evidence. And as I have on the bullet point here is that as these populations separate from each other, they separate in sort of no looking back, right? So the people who crossed the land bridge into North America 20,000 years ago, they lost contact and lost the ability to interbreed with anybody from Eurasia or places farther away. And so they'd accumulate genetic differences just through random chance, random accumulation of mutations or differences, sometimes adaptive, a lot of times not particularly adaptive. And another part of that is that individuals from ancestral populations that are closer together in time, so say the people who stayed in North America as Native Americans, are genetically pretty similar to the individuals who made it to South America as Native South Americans. And those individuals are similar to each other, more similar genetically to each other than either of them would be to somebody in Europe, Asia, Africa, etc. So we'll see some cases when we're looking at humans at differences, sometimes geographically. These relate to the original migrations of human populations for the most part, and of course not counting our modern age where people can cross oceans by airplane and boat, much easier to mix up your genetics these days. All right, so we'd have, back to the genomics and gene discovery conversation, maybe the ability to look for differences among humans that are interesting to whatever we want to study genetically. So this is one approach that you can take to investigating these differences. This is an illustration of a feature that often gets investigated called single nucleotide polymorphism. And it's a fancy term. I guess it's kind of nice. It's got a rather approachable abbreviation that people just call SNPs. So a single nucleotide polymorphism is a single nucleotide difference. That's basically polymorphism. So different kinds. And what it shows is that for the most part, one human to the next would be 99 point, I don't know how many, it's very, very nearly identical between two people. And the places where humans are known to be different from each other, these are the SNPs. So just about everybody, or maybe everybody in the world, is the same for this region, but they're different here, they're different there, they're different there. And these might be meaningful differences that have an association with phenotype, or they might just be inconsequential differences that happen to be differences among people. And as you can see, this is an example of four, say we took DNA sequences from 4 people, that they're not always different in the same ways. And so you might have an explanation for where these differences came up over time. Lots of ability to investigate reasons for the SNPs to exist, and then also how they relate to phenotypes. So if there's a difference between people, it often is due to, it really comes down to something really small on a genetic level. So like I said, humans are very similar from one human to the next for the most part. But once in a while there is a difference, and often it's one of these SNPs, just a single nucleotide change that can give you a pretty big phenotypic change. So I'll spend a bit of time in this semester talking about the SNPs that are known to be associated with eye color. And this is one of them. So this is a resource that will, if you click on the link there, will show you some SNPs across the human genome. And you can see they're identified here. So this is one that's highlighted in the block. It has an A or a G in the human population. So A nucleotide or G nucleotide. And it has this association, this known association with eye color. It's not absolute. It's not to say that everybody who has the G allele always has blue eyes, or the A always has brown eyes. But in a lot of people, and more often, more associated than randomly, the G allele is associated with having blue eyes, the A allele is associated with having brown eyes. So you could do a study to investigate what causes the phenotype by looking at the differences. So this is a map that has 2 kinds of phenotype you might be interested to study. If you take the DNA from people, let's say we have blue-eyed individuals and brown-eyed individuals, if we took DNA from these individuals and compared it to DNA from these individuals, we would look for something that is significantly different and consistently different between those groups that we might be able to use to identify a genetic cause. So we're looking at lots of data compared against lots of data. And by doing that comparison, you might discover, like we were just looking at with the eye color SNP, something that is significantly different in one population versus the other population. So this has the fancy name of genome-wide association study, or GWAS. And this is an approach that's often used. And I think it's pretty cool that we can look at this on the genome level, right? So just casually looking at an individual's, usually tens or hundreds of individuals in one of these studies, and we're looking at individual genomes for each of those people. All right, so just put this together as an illustration of what might show up as a difference. So we've got two examples. We've got a phenotype A on the left and phenotype B on the right, and then a block on top here. This would be, say, one of the SNPs, and then the bottom block would be another one of the SNPs. And if you pool up all the individuals with phenotype A, let's say that's blue eyes, you might get this collection of mostly A's and a few C's, and individuals with phenotype B, still a lot of A's and almost an equal number of C's. So I tallied those up, 42% C's versus A in here, 25% C versus A over here. So we have maybe a little bit more C in the phenotype group on the right, but it's what I would consider a weak association. So they're both sort of C alleles in the minority, and the difference of, what is that, 10%, 20% of difference, versus maybe something a little bit stronger where it's mostly A's in one phenotype group, mostly T's over on the left, mostly A's over on the right, and this would give you maybe more of a strong association. So you can look at a whole bunch of these SNPs and compare them and see which ones have maybe what looks like more of a random distribution of alleles versus something that has more of a firm difference. And I wanted to highlight just, you don't often find something that's so absolute as everybody has one kind in phenotype A group and everybody has the other kind in phenotype B group. But this is still much stronger than, say, a random distribution of T's and A's in this bottom group. So the graph ends up looking kind of like this. This is taking, like we saw already in the beetle example, taking a lot of SNPs across the whole genome. We're in humans now, so 1, 2, 3, 4, up till chromosome 22 here. And sort of a representative set of SNPs across the chromosomes, and a few places here that they've highlighted that are above the, what we call the threshold. So the significance threshold is drawn as this dotted line, and points that are above that are significantly more associated, significantly consistently different between the two phenotype groups. This was a study looking at heart failure and genetic causes for that, and they found across the genome these 3 candidate groups. They got the names for the genes that they think are associated. And on the right here, zooming into chromosome 1, where that ACTN2 gene is located, just highlighting the collection of the SNP locations where those differences are consistent. So a lot of mathematics involved, a lot of statistics, getting an output where it tells you that you've got a significant association between genotype and phenotype at a location in the chromosome. And then you could get in and maybe sequence that region for these individuals and find out what's different as far as being able to produce the enzyme or so on, this ACTN2 candidate gene. And then just a little bit, maybe more of a whole organism kind of question. The one previously, this was looking for genes associated with one phenotype, which was heart disease. They found 3 candidates. This was a study trying to, and there have been several of these, trying to kind of put a genetic map on what somebody's face looks like. So highlighting different parts of the face and gene regions that are significantly associated with those. So you can basically, we're at this era where you can get computers to identify people based on how their face is put together. And we can also use a lot of that technology to ask somebody's appearance, what genes are really associated with that. And there might be thousands of genes, but this is a collection of genes that are being hypothesized as associated with that. On the left here, a whole bunch of genetic regions and the phenotype areas that they're kind of associated with. So lots going on there. One of the interesting applications of this that's been proposed is that you could collect someone's DNA from a crime scene and feed it into your program that understands the connection between DNA and phenotype, and you could actually get a map of what that person's face looks like just based on the DNA they left behind at the crime scene. So pretty cool tools out there for discovering new genes related to whatever phenotype you're interested in. Here's one more, just another illustration going back outside of humans. This is a group of plants that have been found to adapt independently in different populations, adapt to high altitude, and being able to investigate the genes responsible for those adaptations. And they have, again, same thing, these maps where the dots that are high up are supporting the idea of a gene location that is significantly associated with some features. So the variety of features that are found in high altitude plants and the genetic regions that are so far mapping to those differences.